There was a question on one of my previous videos about how oxotrophic mutants are obtained. And in that video I explained how these oxotrophic mutants are used to figure out if a compound has carcinogenic properties or not. And in this video I'll take a closer look at this and explain how these oxotrophic mutants are used for the overproduction of your food or certain compounds. I will focus on the production of the essential amino acid lysine. About 2 million tons is produced each year in large industrial fermenters, mainly used as a food supplement for animals or humans. This little bug is responsible for this yield, Corynebacterium glutamicum. The first question we need to answer is, why do we need to alter these bacteria in order to be able to overproduce lysine? Let's take a look at the biochemical pathway our bacterium uses to produce lysine in the first place. So we start with one precursor molecule, L-aspartate, and through a cascade of different chemical reactions with the use of different enzymes like this one and this one, we obtain 4 different amino acids: lysine, methionine, threonine and isoleucine. But you can imagine that the cell won't just keep producing these amino acids indefinitely, because then the concentrations would become so high it would become toxic. And it's just a waste of energy. You don't need to make more if you don't need more. How does a cell actually manage this? Well, it uses something called feedback inhibition, pointed out with this line here. When lysine and threonine, only these two, increase in the cell, alongside with these other two of course, but only these two are important for the feedback inhibition, they will bind with this enzyme, aspartokinase, which catalyzes the reaction from L-aspartate to this molecule. So when threonine and lysine bind aspartokinase, it cannot catalyze this reaction anymore, and this whole pathway won't happen anymore. And therefore, the production of lysine and these other amino acids is stopped when the concentrations are high enough. But how does this actually work? For that I will take a closer look at how aspartokinase is built up. This is a very simplified version of our aspartokinase, which is an enzyme and thus just a protein complex. And this is the binding site for L-aspartate. So L-aspartate, which is represented by this little cube, can bind our enzyme in the normal conformation when lysine still needs to be formed, and then the enzyme will change L-aspartate to this molecule and it will release it again. But when lysine and threonine is present in high concentrations, this enzyme is more likely to encounter them in the cell. Then lysine will bind here, which is represented by a triangle, and threonine will bind here, which is represented by a sphere. And when this happens, our aspartokinase binding site will modify because of conformational changes. So something like this will happen. And in this way, L-aspartate can't bind this enzyme anymore, and this reaction won't occur anymore. And therefore, all of this is stopped. And the production of lysine is also stopped through this feedback inhibition mechanism. And that's how this works. And now we need to figure out a way to circumvent this feedback inhibition mechanism in order to overproduce lysine. So our cell just keeps producing lysine through this pathway without inhibiting it when the concentrations become too high. And there are several ways to do this, and today I'm going to show one of them, which is most similar to the technique I used in one of my previous videos where I showed how we test if a compound causes cancer or not. The link in the description. Let's have a look at the other side of the board. Try to follow this story about how we become these overproducing mutants, because it's quite interesting how we were able to achieve this just through random mutation, as you will see in a few minutes. So we will make use of revertants for the overproduction of lysine. And a revertant is basically obtained in the following way. We start with a wild type, which is just our C-glutamicum found in nature. We cause a mutation, one mutation, which causes a mutant to be formed. And then we mutate again, reversing the first mutation, So the next mutant actually looks the same as our wild type. That's called a revertant. The second mutation reverses the first mutation. We basically just start with our C glutamicum. We multiply it, make a colony out of it by incubating it for 48 hours in 1 liter of media. Then we need to make the first mutation. We do this just by radiating the cells with UV light, which causes random mutations in the DNA. But how long do we do this and all of this? Let's take a look at this graph here. Here we see the logarithm of the cells per milliliter of media. So here we have 1 billion cells per milliliter, here we have 100 million, 10 million, 1 million, 100, all the way down to 10 and 0 cells per milliliter. What do you think is going to happen when we radiate with UV light? The cells will die because lots of mutations will cause the cell to become completely broken and damaged, and it won't be able to grow and divide, and it just die. So we see actually an exponential decrease in the number of cells. Something like this. And our goal is to actually get only 1 mutation, because eventually you want to obtain a mutation in this little enzyme. But that will become clear in a few moments. And we don't want any other mutations somewhere else which might decrease the viability of the cells. And it turns out that when we kill about 99 to 99.5% of our cells with UV radiation or any method, this causes the most cells with only 1 mutation. Because of course most of the cells will get many mutations and therefore die. Some of them will have only several mutations and very few of them will have 1 mutation. And that's what we want. And most of them occur after about 12 to 15 minutes of radiating with UV. And I said that's 99% of cells dying, so that's around here. Right? So here we have 12 minutes. This is where we stop after 12 minutes of radiating. So only 1% has survived here. With this 1% we work further. We grow these survivors in a new media, 500 milliliters. We grow them so the cells go back up to 1 billion cells per milliliter, and we have a mutant library, which is just a collection of cells with random mutations. Some of them only 1 mutation, others many more mutations, maybe still some wild types left which didn't mutate at all. Just a library combinations of different cells, some of which are also lysine auxotrophic. So in these cells, which are about 1 in 1 million of our mutants in the library, some mutation occurred somewhere in this pathway, doesn't really matter how, which caused that lysine cannot be produced anymore. And this is the cells we need. These are our first mutants we're going to select for. But how do we purify these lysine negative mutants from the lysine positive mutants? Well, we take 1 milliliter of our mutant library. So that's 1 billion cells. From these 1 billion cells, 1 in 1 million are our lysine auxotrophic mutants. So that's 1,000 out of 1 billion cells. We only need to obtain these 1,000 cells. So we add a medium which contains glucose and nutrients. Normal cells should be able to grow in it. But the medium contains no lysine. Cells need lysine to be able to grow, so our lysine auxotrophic mutants, which we want to obtain, won't be able to grow. This is weird, right? But we also add penicillin, and penicillin is introduced into the cell wall of a cell when it divides, when it grows. So when we go back to this graph, I can show it right here. Let's remove the time mark. This graph represents our lysine positive mutants. They die off. Why? Because they are able to grow in this, because we incubate it for 2 days. So we let them grow at 30 degrees Celsius. They start growing, so this remains the same at first. But then penicillin gets introduced into the cell wall and the cells are killed, because penicillin, it's an antibiotic, it kills the bacteria when they grow. So they die off. What happens with our cells which cannot produce lysine? They just stay there, they hang out in the suspension. They don't grow, so penicillin cannot affect them. They actually remain constant. There are 1,000 cells, that's 10 to the third. And it remains constant. And this is how we separate or kill off our lysine positive mutants and only the lysine negative mutants. Right remain. There's only one caveat here. In reality, when the lysine positive mutants die, the cells break open and they've produced lysine, right, to be able to grow. So the lysine comes into the medium. And then, well, after a while, when enough cells have died, it won't be a medium without lysine anymore. So our lysine auxotrophic mutants will be able to take up lysine from the environment, from the cells that have died, and then they will also start growing and die because of the penicillin. So in reality it will be something more like this, and they will also die here. So we stop about here, which is after 20 to 30 hours of incubating. But we have reduced the ratio from almost 1 billion lysine positive mutants versus 1,000 lysine negative mutants to about 1,000. So then with novel screening methods it's pretty easy to separate these two. So now we have our lysine negative mutants isolated. Now we need to obtain a second mutation which causes it to revert back to the lysine positive state. You'll see in a moment why. We do exactly the same again. We mutate with UV light, obtain a mutant library with random mutations, so most of them won't be lysine positive of course. But then we select for the few lysine positive mutants which are present, and selecting for these lysine positive mutants is pretty easy. We just add the mutants to a petri dish with a medium which does not contain any lysine. So only the lysine positive mutants will be able to grow on it and form colonies. And when we now test these lysine positive colonies for lysine overproduction, we see that about 1/3 has lysine overproduction. But this is weird, right? We just have a wild type, which is lysine positive. We do a first mutation, select the lysine negative mutants from that, and on these lysine negative mutants, we reverse the mutation back to lysine positive, as suddenly 1/3 of the cells have lysine overproduction. To understand why this happens, I'm going back to our aspartokinase enzyme to show you an example on how this could actually happen. So, we have our enzyme in the native state, which is without lysine or threonine bound to it. We induce a first mutation to a lysine negative mutant. That would imply that, for example, our L-aspartate can bind this anymore. For example, here there is a conformational change because of changes in the DNA which result in changes in our protein here, and this cannot happen anymore. So this would be a lysine negative cell, right? Because the aspartokinase it forms has this structure and L-aspartate can bind more, and therefore this can't happen anymore and lysine can be produced, right? Now when we mutate again and obtain a lysine positive mutant, this has to disappear, right? Because we need this allosteric center to be functional, so our L-aspartate can bind again. But in 1/3 of the cases we see that when you reverse this, something here nearby this allosteric center causes a mutation in that allosteric center. And what does this imply? Lysine cannot bind anymore to our aspartokinase. This mutation could also take place here for threonine, by the way. Doesn't really matter, but it has to be close to this allosteric center. And the fact that lysine can't bind anymore means that this feedback inhibition can't happen anymore, because if you remember, when lysine and threonine binds, both of them have to bind. That causes feedback inhibition, preventing our L-aspartate to bind our enzyme. This causes the overproduction because feedback inhibition is gone, and this just keeps happening. And that's how we can get very high concentrations of lysine in our cell for the production in the industry and eventually also the food on your plate. I really hope you enjoyed this video and found it a little bit insightful on how we actually do it. Subscribe if you haven't already, it really helps me out, and I see you next time.